Hello, my name is Mahesh. I'm currently a computer science and bioengineering major at Berkeley.
My name is Haik. I'm a student at UCLA studying computer engineering and math.
And hi, my name is Karthik. I'm a student here at UC Berkeley studying CS and business.
Yes. I've been a biologist for the past 7 years and worked in my first biology lab in seventh grade. Many electronic lab notebooks are revolutionizing the way that we can store data and analyze our data online. However, with these recent advancements, we all know that biology is the next field that's going to be radically changed, and we want to be at the forefront of that. That's why we created BenchBrain, a program where it's able to have access to your entire laboratory notebook, which I'm going to open right now that we've built. So it has access to your entire laboratory notebook, all of the data, all of your results, your hypotheses, your sequences that you can see right here. Has everything, has all the context that a researcher would have. But it also has AI analysis enabled. So what we are doing is we've talked with researchers. This hackathon, we talked with a lot of researchers, and we talked to them and asked them, what do you need? What problems do you have? And some of the biggest problems were having to do a lot of research and having to go through multiple different papers and spending hours and hours of time on that and connecting their research to these papers and doing statistics and statistical analyses, because these are guys that are focused, that have their PhDs in biology and chemistry, and they have a difficult time running data science tools and statistical tools. That's why we created an AI researcher that basically acts as another researcher for them and is able to find breakthroughs and novel findings in their research in their electronic lab notebook.
All right. So, going over how it actually works. So, the researcher is an agent that we build using Letta. Now, for the sake of the demo, to save time, we have pre-filled this out. So, you could imagine a researcher is in the middle of research and has filled out all these notes, okay? And we've also already had the analysis occur, but you could just imagine we click the analysis. To show what actually happened in the analysis, we can go to Letta. So, Letta has a few parts to it. So, first of all, with Letta, we use this thing called Bright Data. Bright Data lets us scrape the entire web for any relevant research. This is essential because any breakthrough, any statistics it makes, all of that is based off of online data. So this is an example of when the researcher enters something to the notebook. If we scroll down, as you can see over here, Bright Data is searching online for useful things. We can see over here, pKa phosphorylation and more. Okay, and the Letta agent is storing all this into its archival memory, which it can do RAG over, and basically anytime it needs it, it can get the relevant data. And we continue, and then when we click Analyze, we have the agent basically return a JSON output that we convert into the actual UI for the researcher. So we click Analyze, now we can go over here. So right over here, we have an entire breakthrough summary for what the researcher can do next. We have a recommended protein edit, we have an expected outcome, and we have links everywhere. This is all from Bright Data. So you click this, you see the relevant research. And we even have a nice graph over here that you can interact with, and it tells you what each protein, what each node does for this breakthrough, and all the interactions. And then we also have the agent run statistical tests. So we can see the statistical tests it wrote. We have nice visualizations over here, and we can see all the math for the researcher to double check. And lastly, we have cross-reference data, and this shows all the links it has used.
Thank you so much.